Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture. Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture.
Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture. Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture.
Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture. Puzzled by your sequencing results? You've seen the incomplete picture of short reads.
Very good morning to all participants. Welcome to the third and last day of the 4th International Conference on Molecular Biology and Biotechnology, in conjunction with the 27th Scientific Meeting of the Malaysian Society for Molecular Biology and Biotechnology, with the theme Beyond 2021, Building Sustainable Science. First and foremost, I would like to introduce myself. My name is Sok Mui from University Technology Mara, and I will be the MC today. On behalf of the organizer, I would like to extend my warm welcome to all the participants to the 4th ICMBB 2021. Before we start, I would like to remind all participants on the house rules. Please mute your video and audio during the conference. You'll be given instruction when you need to turn on your video. If you leave the room and want to get back in, or you want to move to another room, you have to click on the link in the program book and you can move in. If you have question to ask during the Q&A session, type your question in the chat box and to whom it is to be addressed. Please remember to fill in the feedback form on this conference to allow us further improve in following conferences. The link to the feedback form is in the chat box. Ladies and gentlemen, we will start the concurrent session 3 at Hall 1 and Hall 2 at 9:10 a.m. The current hall is Hall 1. If you would like to listen to the talks on medical biotechnology and molecular biology, please remain in this hall, Hall 1. If you are interested in microbial biotechnology and molecular biology, please go to Hall 2. To do so, you may click the link in the program book to go to Hall 2. Ladies and gentlemen, please let me introduce our chairperson for this session, Associate Professor Dr. Siti Hamimah from University Technology Mara. Dr. Hamimah, the screen is yours.
Thank you, Dr. Sok Mui. Assalamualaikum and very good morning to all participants and professors, speakers, and all attendees. Before we begin, let me briefly introduce our fifth preliminary speaker for this conference. Professor Dr. Muhammad Effendi is a senior professor in the School of Fishery and Food Sciences, University Malaysia, Terengganu. He is a member of Institute of Marine Biotechnology and Institute of Tropical Aquaculture and Fishery both in University Malaysia, Terengganu. He is working on understanding the potential of mucosal immunology in small ruminants in protection against respiratory bacterial pathogen. Apart from immunology, his research has actually expanded in searching for bioactive compound from traditional herbs, as well as aquatic resources for health purposes. Example, speeding up the wound healing process, reducing hypercholesterolemia and the side effect of compounds to the host cells. He has published more than 70 research articles and actively collaborating with many international institutions. Without further ado, I would like to invite Professor Muhammad Effendi Abdul Wahid to deliver his talk. The screen is yours, Prof.
Thank you, Professor Bima. Can you see my slide?
Yes, Prof.
Okay, thank you, Madam Chairperson, Professor Dr. Siti Amimah. Aquaculture has been identified as one of the main sectors in coming years as part of a global solution to the rapid depletion in the world's natural fish stock. In Malaysia, the government is looking to expand the agriculture sector, specifically the aquaculture industry. However, aquaculture industry will not sustain if the waste from the activities are not well managed. My presentation today is about the potential from aquaculture waste as biomaterial for biomedical application, that is chitosan hydrogel biomaterial from fish scales for topical wound healing application. Frankly, I don't think everybody here is familiar with the term fisheries and aquaculture. Perhaps one might think both terminologies have the same meaning. So let me start off my presentation with the brief definition of this term. Fisheries are solely related to catching wild fish or raising and harvesting fish through aquaculture or fish farming. Most of the fisheries activities is happening at the sea. Aquaculture is also called fish farming, and it involves the natural and controlled cultivation of shellfish, fish, and seaweed in freshwater and marine environment. Food and Agriculture Organization (FAO) stated that aquaculture surpassed wild capture as our main source of seafood for the first time in 1970s. Since 1990s, aquaculture production has more than tripled. And today, more than 200 species of fish and seafood are raised in farms. The increasing demand for aquaculture to fulfill the demand for seafood protein is expected to continue until 2050. Numerous reports from global research agencies have all been reporting the devastating impact of fisheries-related waste that directly affects the biodiversity and mankind. Estimations have shown that more than 50% of fish tissues end up being discarded as waste, which includes fins, heads, skin, scales, and viscera. I think most of you like to eat head curry fish, right? For instance, let's take tilapia as a subject. Since the topic today is about the scales from this fish, tilapia is considered as marine chicken in seafood industry due to their popularity. Not everyone here is a fan of this fish, but they are considered as one of the most popular fish in aquaculture. 80% of the fish consists of their flesh, whereby the food industry utilizes 100% of the flesh for their business. For example, for fillet, burger, and fish balls. 12% of the fish is composed of internal organs Some are edible and 60% of them can be used as fish meal, while another 40% will be discarded. The remaining 8% is the skin and scales, where the sector used about 20% of them as biomaterial and 80% was wasted and discarded. Many review articles have shown that fish waste materials to be highly utilized in extracting fish gelatin due to its varied biomedical and nutraceutical applications. Fish gelatin in most cases are accepted more readily than mammalian gelatin due to ethical and religious point of view. As you know, Muslims cannot consume gelatin from pig or non-slaughtered animals. Hence, many studies have now shifted its focus on optimizing the extraction methods for increasing the recovery of gelatin from fish waste, particularly fish skins, bones, and scales. Fish scales, which constitute of 8% of the total fish collagenous waste, is made of biocomposites of highly ordered type 1 collagen protein and hydroxyapatite. So the objective of this presentation, the objective of this study was to produce chitosan hydrogel using chitin extracted from tilapia fish scales, and secondly, to determine the ability of chitosan hydrogel to heal wound in Rattus novigicus. For the production of chitosan hydrogel, tilapia fish were obtained from Como River in Kenyir Lake through the Institute of Marine Biotechnology, University of Malaysia, Terengganu. The fish were euthanized in cold water with ice, and the scales were then removed, collected, washed and dried in oven at 60 degrees Celsius for 24 hours. The dried scales was then mixed with 1 molar hydrochloric acid and magnetically stirred using hot plate stirrer at ambient temperature for 4 hours. The resulting solid was washed with filtered running tap water till neutral and then dried in oven at 60 degrees Celsius for 24 hours. For deacetylation, hydrolysis of acetyl groups was accomplished by reacting 50% sodium hydroxide with the demineralized scales at 110 degrees Celsius for 1 hour. The chitosan products obtained was washed with distilled water, followed by deionized water to neutrality using sieve before dried in ambient temperature. The dried samples were purified with methanol and ethanol before dried in oven for 24 hours. For the preparation of chitosan hydrogel were prepared with the formula of 1% chitosan powder in 1% acetic acid. The solution were filtered and poured into petri dishes to be left overnight at room temperature. The resulting chitosan films were then mixed with 3 ml sterile water for hydrogelation and stored under refrigeration at 4 degrees Celsius until use. For the experimental design, a total of 18 clinically healthy matured male Swiss albino white rats were equally divided into 2 groups, the control and the treatment groups. Wound incision was treated with sterile punch biopsy, and the control group was not given any treatment, while the treatment group was treated with chitosan hydrogel 2 times per day. Commercial pellets were given daily at the rate of 250 grams per animals per day, while water was provided ad libitum. A total of 3 rats from each group was sacrificed, and then surrounding wound tissues areas were excised for histology evaluation. On day 7, day 14, and day 21 post-wounding, the granulation tissues were removed for further analysis. The efficacy of treatment using chitosan hydrogel was evaluated based on the wound contraction and re-epithelialization time. The changes of wound surface area were observed daily to keep track of its wound healing progress. The excision wound margin was traced using transparent sheet and recorded into graph paper to measure the surface area in millimeters square until wound healing process completed. Wound contraction was expressed as reduction in initial surface of wound in percentage as shown in the following equation. For results and discussion, production of hydrogels were less consuming in terms of time, energy, and costing. The successful extraction of chitin from tilapia fish gills can provide an alternative resource for the production of the highly needed chitosan-based biomedical product. This figure displays development of wound surface areas for control and treatment groups from day 0 until day 21. Formation of blood clots and scabs can be observed in both groups. On day 3, with relatively greater contraction and smaller wound surface area in wounds treated with chitosan hydrogel. On day 7, the scabs from control groups were still fresh, while in the treatment group, the scab on the upper wound has become very hard until the scab from lower wound, while the scabs from the lower wound has dried and fallen off. The treatment group continues to show faster recovery with full closure of wound and much hair growth on day 17, while scars can still be seen in control group even on day 21. The moist wound healing environment promoted fast epidermis migration for accelerated epithelialization, as it was easier for the epidermal cells to migrate over a moist surface than under a scab in dry wounds. The hydrogels was compatible with most living tissue with its higher water content and viscoelastic nature that minimizes damage to the surrounding tissue. Chitosan has helped to block nerve endings, thus reducing pain sensation. This figure shows the progress of wound healing histologically, where the treatment group showcased faster improvement in comparison to the control group. On day 7, significant stimulatory effects on granulation and capillary formation can be seen in treated wounds. The control wounds, meanwhile, collapse without thick scabs formation unaided by chitosan hydrogel, like in the treatment group. On day 14, it can be seen that the treatment group underwent remodeling phase with accelerated granulation and aggregation of collagen fibers towards the wounded area in comparison to that of the control group. On day 21, treatment group demonstrated complete re-epithelialization and mature collagen deposition with matrix formation and new hair follicles growth. Wound in control group, however, was still in proliferation phase, exhibiting less collagen deposition and granulation below the wounded area. It was not yet completely covered with new epithelium, and there is only moderate number of inflammatory cells present in the upper dermis. This figure shows that wound in mice treated with chitosan hydrogels generally heal faster than those in the control group. On day 3, for instance, the percentage of wound closure in treatment group, which is 57.48%, has already been twice higher than of the control group, which is only 29.36%. On day 6, 3/4 of the wound surface areas in treatment groups were closed, while the wounds in control groups contracted not more than half of their original size. Full wound closure of epithelialization was attained by treatment group on day 15, 100%. Okay. While the control group took about another 1 week to obtain the same result. So this is very important if the patient has diabetic, because diabetic wounds need to be healed faster to avoid bacteria or any bacterial contamination into the wounds. These enhanced caps provided a temporarily protective barrier and minimized the infection risk, allowing sufficient time and space for smooth progress of granulation and angiogenesis. The contraction, which is under the control of transforming growth factors and other cytokines, is aided by collagen in the extracellular matrix in locking the tissues in locking the cells in place, thus augmenting the contraction process. This table, this figure shows the percentage of collagen density in control group without any treatment, chitosan hydrogel treatment group, and normal collagen density group. The collagen density in the treatment group showed relatively higher percentage compared to the control group at day 7, 14, and 21. In fact, on day 21, the treatment group achieved 100% of collagen density, which was akin to the normal collagen density level in skin. Whereas the collagen density achieved by the control group was still less than 100%. This table shows the collagen density was significantly different between control and treatment group at days 14 and day 21 post-wounding. Cost-saving production of chitosan hydrogel using chitin extracted from tilapia fish is proven to be a good alternative resource for the relevant biomedical products. The wound treated with chitosan hydrogel has been proven to heal better compared to the control group without any treatment. The treatment can be applied easily via swabbing the chitosan hydrogel with cotton buds onto the wounds, and all mice did not show any signs of rejection or distress to the moistening dressing during and after the treatment, rising to the advantage of full adherence with greater interaction between wounds and the healing agent. Chitosan hydrogel work very well in absorbing excessive wound exudates to facilitate healing and enhancing scab formation, providing a temporarily protective barrier from infection. The wounded skins generally attain full wound closure with accelerated epithelialization and over 95% of collagen density for remodeling within 2 weeks post-wounding. The contraction became the dominating factor of the wound closure, occurred with wound shrinkage by pulling edges of the wound towards the center, aided by collagen in the extracellular matrix in locking the cells in place. The accelerated wound healing process stimulated by chitosan hydrogel with its association in collagen production, fast epithelialization period, and wound reduction suggests a significant positive effect as an effective wound healing agent in the future. Thank you very much.
Thank you, Prof. Afandi. I hope all the audience and speaker can still can remain in this hall till 11 a.m. All the question and Q&A session will be held at 11 a.m. Now we move on to our second speaker. Let me briefly introduce our second speaker for today, Associate Professor Dr. James Badger-Wink. He is originally from UK. Dr. Wink completed his PhD and training of postdoc research at the University of Sheffield, UK, where he worked on innate immunity and vaccinations. From here, he became interested in the regulation of B-cell responses and moved to the laboratory of Professor Simon Sakaguchi in Immunology Frontier Research Center, IFRAC, Osaka University, 2010. While there, Dr. Wing research have focused on the control of B-cell responses by Treg, regulatory T cells, and their subgroup. In 2019, Dr. Wing have formed the Laboratory of Human Immunology, focusing on single-cell immunology as a new group in IFREC Osaka University. He is currently working on the use of mass cytometry to explore the diversity of Tregs and other cell types in a variety of settings, such as autoimmunity and infectious diseases. And currently, Dr. Wing is working on understanding responses, immunity in COVID-19. And without further ado, I would like to pass the screens to Dr. Wing to present his research. Yes, please, Dr. Wing.
Thank you. Thank you very much. So thank you for the invite. Good to see you again. So I'll just quickly get on to sharing the screen. One moment. I hope you can see that okay?
Yes.
Yes. Okay, good. So yes, thank you very much for letting me talk today. And basically, so I'll be talking a little bit about single cell analysis of COVID-19 patient samples by mass cytometry. So essentially, what we're doing here, so it's nothing so complicated in terms of sample collection. So we're taking peripheral blood mononuclear cell samples from COVID-19 patients. And in the data we're showing today, essentially there's a total of 100 different kind of samples of 40 healthy controls and 60 COVID-19 samples across various states of severity, the most common being severe, which is, this is defined by the kind of a WHO ordinal scale. And so the severe patients here are kind of under kind of intubation in intensive care. And we also have some follow-up samples, which is some of these patients, but 20 to 60 days later, when most of them have recovered at that point. And here we've got a total data set of around 15 million cells. And just to explain what we're kind of doing here from a methodological point of view. So as mentioned, we're using mass cytometry, which is this technique you may also know is called a cytoff sometimes. So essentially, this is similar in kind of concept to flow cytometry, with the kind of the main difference that we replace, your classical fluorochrome labeled antibodies with metal labeled antibodies, essentially. And the internal, workings of the machine are somewhat different, but the basic point is you're able to collect, cytometry data on around 30 to 50 different markers per cell. And this is the main benefit of mass cytometry above kind of other techniques, just the ability to collect more markers per cell, essentially. And in terms of, the design of what we're doing in terms of staining of the cells, so this is basically, immunology based stuff. So we're staining these samples with a 50 parameter mass cytometry panel. So this is 50 different proteins kind of being measured on every cell. And we have a combination of lineage markers to identify major populations, chemokine and cytokine receptors, various transcription factors, and then varied kind of activation like exhaustion and naive markers and such like, to give us a kind of a relatively broad overview of the immune system in these kind of during infectious disease. So essentially, here we're looking at just the kind of the global overview of our CD45 positive cells, which is kind of pan immune cell marker. And of course, in this approach, we're generating a lot of complex data, and then we're using various techniques to kind of, bring it down to an understandable level. And one of the main ones being dimensionality reduction. So we're taking, our 50 or so dimensional data, 50 markers, and boiling it down to a two-dimensional structure. In this case, we're using UMAP to do that. There's a few different ways of doing it. You can use Tisney and such like, but we find UMAP most suitable in most cases. I mean, essentially, this little graph to the side here, you can hopefully see. The colorations are the different clusters we've identified. So we also use algorithmic clustering, in this case, a technique called FlowSum, to identify, major populations. And then I've just, this is just manual annotation of our interpretation of what these different populations actually are. And here as well, you can see some of the major markers which have gone into this identification. So this is not the full panel, but just a couple of examples of markers which help us to identify our major populations. So for example, here, very simple one, we've got CD3, which of course expressed by CD8 and CD4 T cells. And here we have CD4 on our, so here is our CD4 T cell population. And then when we get into the issue of, I guess the key question is, what's actually changing, in different, different severities of infection. So here essentially, so we have our major lineage populations mapped out by the UMAP. And then here, just using box plots, we can show this is just proportional analysis of what cellular populations are changing, at different levels of severity of infection. And just to explain this, because I use the same layout throughout, essentially we've split our healthy controls actually by age into a kind of a younger, under 50 years old and over 50-year-old groups, because actually most of the COVID patients tend to be over 50 years old. And then essentially here we will have moderate infection, severe infection, critical infection, and then the follow-up samples. So as you can see here, we have this big change in classical monocytes. These are kind of fairly normal kind of antigen-presenting cells you'll find in blood. These are very much increasing proportionally during infection, and particularly with severe patients having severe and critical patients having high levels. On the other hand, so something goes up, other things have to come down. So we see our non-classical monocytes being actually very severely kind of lost in the blood during infection. And that appears to be transient because at this follow-up stage, you can see they've kind of rebounded. And essentially for the other populations, one of the other big changes is plasma cells. So this is our antibody-producing B cell population. That we're seeing very much increased during different stage of infection. So again, increased particularly during kind of severe, in severe cases. And then, of course, so we can see these big kind of lineage populations. We see all the significant changes like here. We're using logistic regression by EdgeR for the statistics here, but I'm not going to get into too much detail other than say, because we've got a lot of samples here, if it looks different, it probably is significantly different as well. And then essentially, because, so as mentioned, some of my research interests are around some particular subsets, such as kind of T follicular helper cells, regulatory T cells, etc. So we're using a reclustering approach for kind of greater depth of analysis. So this is conceptually fairly simple. All we're really doing is we're taking our initial kind of big picture lineage analysis, and then we're identifying, for example, CD4 cells. And then we take those cells and then we recluster them using essentially the same approach. It's the same data set. We might change the exact markers used for clustering a little bit because, as you can imagine, some markers are better for other cell populations. And this allows us to get into the into the kind of fine details of these various cellular populations. And today I'm mostly going to focus on the CD4 and regulatory T cell areas. So in terms of our CD4 subset analysis, so essentially here again, it's a similar conception. We have our UMAP dimensionality reduction of our kind of CD4 populations, and then our variously identified clusters, which I've kind of annotated here. And just to give you some orientation for some of the markers which are going into this kind of identification, here, for example, we have CD45RA, which is a very much kind of classic naive T cell marker. Here we have CCR7, which is retained in naive and also down into kind of central memory regions of the cellular populations. And here we have CD27, which is lost in the relatively terminal effector stages of CD4 development. And also, as well as this kind of main structure, we can see there's several kind of little islands of cells coming off the side here. And so, for example, here, this one, which is actually kind of far away from the rest, this is our kind of relatively cytotoxic CD4 population, defined primarily by granzyme B expression and also CD57. This, as you can also tell from the loss of CD27, is a fairly terminal population as well. Then we have CXCR5, which is identifying our T follicular helper cells, and FOXP3, which is the kind of main transcription factor that we would use to identify, the suppressive regulatory T cell or Treg kind of a subset, which we are often interested in. And then again, so once we get into the issue of, so actually what's changing, between healthy controls and people in various kind of severities of COVID-19 disease. One of the big initial changes we see within CD4 is actually kind of a shift. It's a kind of a subtle shift, but it's quite a large proportional shift within the naive CD4 T cell populations. So actually, we're seeing this decrease in sort of just what I guess you call kind of normal naive CD4 T cell, and an increase in particularly a CXCR4 high naive CD4 population, which has also lost some other markers such as CD31, which is kind of recent thymic emigrant marker. And this is looking like a kind of, broadly speaking, still naive CD4 T cell, but at kind of a later stage of their development. So they're kind of shifting kind of along the developmental track during infection. And it's also true that we see, also a bit of a shift down into kind of central memory region being increased during severe infection. And then, as you might well expect, so we see broad, cellular activation and proliferation within CD4 T cells. Particularly here, you can see, kind of going up with different kind of severity of infection, Ki67 positive population. And Ki67 being a marker of recent proliferation. So these are essentially, CD4 T cells are actually actively proliferating within the blood. And we also see some phenotypically quite suppressive looking CTLA-4 high CD4 T cells. And CTLA-4 is a molecule we know have very important kind of suppressive functions. It's normally associated primarily with regulatory T cells. But these cells, we don't actually think these are regulatory T cells per se, but they're just relatively suppressive looking conventional CD4 T cells. And then over here, our island, here we have our actual regulatory T cell, and we do see some shifts in their kind of proportion, such as a loss of naive Tregs. But actually, I'll go into more depth in that in a couple of slides. And then essentially, one of the things we're doing to kind of get a better idea of, what's happening to these kind of cellular populations is what's called trajectory analysis, which is just, it's just kind of a simple way of laying out the cells in a kind of a relatively ordered fashion. And here we're using a technique called Pagetree, visualized here by Force Atlas 2 algorithm. And essentially, we're starting from our CD45RA positive, CD31 positive recent thymic emigrants. So this is kind of the most naive T cells, which are, available in the blood. Taking that as a starting point and then seeing how the cells differentiate away from there, or at least their trajectory away from there. So as mentioned, we do see a kind of a relatively COVID-specific branch still within the naive region, but like a kind of a slightly later looking kind of naive population, which is kind of differentiating there. I mean, of course, as you kind of expect, you see going, differentiation through from naive to kind of central memory in our classic kind of TFH, TH1, etc. populations, going through into this quite terminal granzyme B, relatively exhausted looking CD4 T cells. But also, particularly within COVID-19, we do see this extra branch coming off to the side here, which is this very much highly proliferative, but also very suppressive looking CD4 population, which is of some interest to us. And then, so to go even further down the kind of, the rabbit hole, as it were. So of course, so we have 15 million cells here. So we're able to get right down into the details of some of these fine cellular populations. So as noted, I'm quite interested in regulatory T cells, and we do repeat this, this trick essentially of reclustering and reanalysis of our regulatory T cells in fine detail. So essentially here, this is, so our regulatory T cell, these are all FOXP3 expressing suppressive CD4 T cells identified. So Treg, because they come out of the thymus as a kind of a relatively distinct lineage from other CD4 T cell populations, they do, within their own kind of population structure, they do kind of cover the full range of going from sort of naive or central-like cells into highly activated kind of effector memory-like or proliferating populations. And again, here, this is just a selection of a couple of the markers which we used to identify these various populations. And like conventional CD4 T cell, we have this sweep going from CD45RA, relatively naive looking, CCR7, gaining CD95 as they become more activated, and then also like a few other markers which help to give it some structure. And in terms of what's really changing during infection, I mean, you can see also the density plots here, but in terms of, the main kind of proportional changes. So as previously noted, we do see this quite clear decrease in naive regulatory T cells within the blood of COVID patients. We also see a decrease in T follicular regulatory cells at the same time. And then kind of, again, as expected, we see increasing activated cells. But actually, once we get right down into the details of it, it turns out that different regulatory cell subpopulations have slightly different distributions, and particularly, so it's not quite as simple as just general proliferating regulatory T cells are correlated with more severe infection. So actually, we find, so these are a couple of previously described subpopulations of effector Tregs, so HLA-DR high and CD38 high cells. And actually, it's really the CD38 expressing regulatory T cells which separate, for example, severe and critical infection. So if you can look here, you can see actually this particular subgroup is particularly associated with people in a critical condition. So actually, we can pretty much tell the difference between different severities of COVID-19 infection by looking at the regulatory T cell subsets. And then, so essentially, just, in the interest of time, I don't attempt to cover it in any detail here, but we are looking, so as mentioned, we've been looking in great depth at these particular populations. But of course, we're also taking trying to get a broad sweep of the immune system, looking at other subpopulations such as TFH, but also getting into, our CD8 subpopulation analysis, B cells, NK cells, and generally speaking, the myeloid cells. So just, as one major feature of this, for example, we do see, and this is expected from the literature at this point, because we're not the first person to look at some of these things. So for example, we do see within kind of our classical monocytes, which are broadly speaking expanding, we see this big increase in a HLA-DR low population. So they're losing some of their antigen presenting capacity. And this, again, this is very much associated with kind of a more severe and critical infection. And then essentially, so this is, this kind of too much data to fit on the slide, but just to boil this down, so here we're doing correlation analysis. So actually we're taking all of these, all of these different kind of subpopulations that we're looking at, and then we are having a look at, how they correlate with one another and how they correlate with disease severity, for example. And as you can imagine, so these changes to even, things like subpopulations such as Treg, I mean, they don't happen in isolation. It's really a kind of a whole broad sweep of the immune system moving in a certain direction. And we do find that various cellular populations correlate with disease severity and also one another. So this is some of these populations, and this kind of, this is a Spearman rank correlation. And essentially we're seeing positive correlations between both, disease severity and these populations, and also within each other. And these are not necessarily closely related populations. So we're now in the process of trying to work out, which ones are driving the others, or is it, is it a central kind of a factor which is driving the expansion of all these disparate populations? So we have, for example, our CD38 Tregs, some CD69 positive activated NK cells, these HLA-DR low monocytes, and also plasma B cells are all kind of going up as a group. Whereas on the other hand, things like naive CD8, non-classical monocytes, various dendritic cell populations, and also some naive and memory B cells, generally speaking, being reduced. And now this is ongoing work. So now we're in the process of really trying to work out, which are the actual central factors driving, this broad shifting within the immune system of COVID-19 patients. This is ongoing work, so conclusions are a bit like preliminary, but broadly speaking, what we can say is that, as expected, this is a very severe infection, and we do see large phenotypic shifts in proportions of various cellular populations. Of interest to us, of course, is that when you look at regulatory T cells and their activation status, actually you can see real differences even between, subgroups of quite severely infected patients. And then, as mentioned, these populations don't go up in isolation. They kind of go up in a whole group of cells across various lineages. They're all kind of going up together. And at the same time, other cells are kind of going down proportionally. So just quickly, acknowledgement to the members of my lab for helping out all of this work. And of course, a big thank you to all the patients who gave consent for their use of the samples. And as you can imagine, there's a lot of collaborators involved in actually the sample collection. There's too many to kind of name them individually here, but just like to, mention, of course, this is not just, one person doing this. There's a whole lot of people doing this work. So I think with that, I'll hand you back to Hamima. And I think there's questions later on. So I'll look forward to answering any you have in the later Q&A session. So thank you very much for listening.
Thank you, James, for sharing an interesting hot from the oven research finding on COVID-19. I know the audience have a number of questions for you, James. I hope you can remain till 11 a.m. for the Q&A sessions.
Yes, will do. Thank you.
Thank you, James. So we are taking 5 minutes break, and we will continue at 9:55 with all the presentation from our participants. Yeah, so we will taking 5 minutes break, and I hope I see you at 9:55 for our oral presentation by participants.
Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture. Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture.
We are back. Morning again to everyone. Salam sejahtera. We are starting with our oral presentation from participants. Our first presenter is Ms. Noani binti Ahmad. Ms. Noani will be presenting her research finding entitled Cool Gas Spray Hydroxyapatite Coating with Thermally Grown Interlayered Titanium Oxide. Ms. Noani, are you ready?
Yes.
Yes.
Okay. Okay, thank you, Chairperson, Associate Prof. Dr. Siti Hamimah. Assalamualaikum and a very good morning to everyone. So my name is Noani binti Ahmad. Today my presentation title was Cold Gas Spray Hydroxyapatite Coatings with Thermally Grown Interlayered Titanium Oxides on Titanium 6 Aluminum for vanadium. Oops. Okay, so this is my content outline for today's presentation. So increasing older people increase the demand for titanium 6 aluminum for vanadiums biometric implants to enhance their quality of life. And HA coatings has been introduced. HA coating has been introduced to improve the bioavailability of titanium 6 aluminum for vanadium, as titanium 6 aluminum for vanadium has lacks its bioactivity. However, the common plasma spraying used to coat the HA produce amorphous HA due to their high temperature workings, and titanium oxide was introduced as interlayer oxide to reduce the amorphous HA coating and also enhance their adhesion strength. Therefore, in this study, a low temperature working, which is the cool gas spray, was used for coating to retain the HA crystallinity. And thermally grown titanium oxides was also introduced as interlayer oxide to reduce the thermal mismatch of HA and substrate that led to the HA delamination and cracking. So this is the first reporting of the cool gas spray HA coating on the thermally grown interlayer titanium oxides on titanium 6 aluminum for vanadium. So this is my experimental research design. So for objective 1 is to produce and characterize the titanium oxides by thermal oxidation. And objective 2,
So can you turn on your video? Sorry.
Okay, start the. Can you see me?
Yes, we can see you.
All right. slideshow. Okay. So for objective 2 is to code and characterize the HA coating on the untreated and thermally oxide interlayer titanium oxide by cold gas spray method. So for the methodology, it's divided by 2. First, the thermal oxidation of titanium 6 aluminum vanadium and their characterization. And second part is the HA coating and also their characterization. So for thermal oxidation of titanium 6 aluminum vanadium, first, the sample was sand grade with 260 grades silicon carbide paper, and then the sample was treated with a medium temperature in a furnace at a normal atmosphere environment. The medium temperature was chosen so it will provide a very good properties of the titanium oxides. And then the thermally grown interlayer oxide, titanium oxide, was characterized by their phase composition and surface morphology and elemental acidities by FPCM EDX, and the substrate hardness was determined by viscous hardness with 5 indentation, and the value was averaged out. And then for cold gas spray HA coating, the spraying parameter was according to the Hasnayati. First, the substrate was preheated at 600 degrees Celsius to provide the plastic deformation for the particle to develop on the surface. So it is known that oxide layer may be produced because of the sample preheating. However, the degree of the oxidation with the chamber of cold gas spring is impossible. And then the HA particle was accelerated by the gas that remained at a room temperature to retain the HA crystallinity. And then the HA particle will impact the surface substrate to coat the substrate with the HA powder. And then the HA coating was characterized by the phase composition and surface morphology and their elemental analysis. So for the result and discussion, first, this untreated and thermally grown titanium oxides was characterized by SRD. As you see here, there are color variation as the oxidation temperature was increased. The bare titanium 6 aluminum vanadium was a metallic color, and increasing the temperature, the sample color from dark light brown to the darker brown. So all samples dominate by the alpha titanium phase, and the it is indicate a very thinner oxide layer was produced, and oxygen diffuse layer was found in all samples, and aluminum vanadium oxide peaks are not detected due to their low composition of vanadium and aluminum. And anatase titanium oxides was formed at 500 degrees Celsius and 600 degrees Celsius thermal oxidation temperature. And for the surface morphology, it indicates all the same surface morphology for all temperature. It is because of the sand grid growth before the thermal oxidation performed. So increasing the thermal oxidation reduced the aluminum's composition. However, the vanadium composition was not detected by the EDX, and the percentage of oxygen also decreased, increasing the temperature of oxidation. So increasing the oxidation increased the hardness of the substrate. However, at 600 degrees C, the hardness was low due to the different oxidation rate of polycrystalline alloy and their formation of oxide, and also because of the microstructure in homogeneity. And for the phase composition of the HA coatings, is if you can see here, the coating shows the same phase, HA phase as the HA powder. It is indicate that cold gas spray preserve the HA crystallinity, and alpha titanium peaks here was detected due to the X-ray penetration to the substrate of the samples, and it is also indicate that thermally grown interlayer titanium does not give significant difference in HA peak crystallinity. So for the surface morphology of the HA coating, if you can see here, the coating was the coating was dense, okay, and there are cracks observed in all samples except at 550 degrees C, and based on the EDX analysis, increasing the thermal oxidation increase the calcium deposition and decrease the oxygen percentage. Okay, this is the dense coatings was due to the high particle impact of the HA powder, and it also indicate that HA particles were bonded together during the spraying. And as for the conclusion, the thermally grown interlayer titanium oxides were produced by thermal oxidation, exhibit the formation of oxygen deformation with mixed oxides and alpha titanium as a predominant phase, and the surface morphology grown the same morphology throughout the thermal oxidation temperature, and the hardness was increased when oxidized compared to the untreated. And the cold gas spray provide the dense HA coating with or without the thermally grown interlayer titanium oxides and remains the good crystallinity of the HA. Therefore, the thermally grown interlayer titanium oxide does not have an effective effect on retaining the HA crystallinity. So further investigation needed to be understand for the effect of thermally grown interlayer titanium oxides on HA properties such as coating adhesion strength and also coating dissolution rates. So this is my reference. So thank you for listening.
Thank you, Ms. Nur Aini.
Yes.
We now move on to our second oral presenter. Can you please unshare? Yes, share the second presenter slide. Our second presenter is Ms. Chi Chiok Woo from University Malaya. Are you ready, Ms. Chi? Ms. Chi will be presenting her research finding entitled Morinda for Morinda citrifolia as a potential anti-proliferative agent on colorectal cancer cell lines.
Hi.
You may present now, Ms.
Yeah,
Chi.
can you see my screen?
Yes.
Okay. Thank you, host. Good morning, everyone. I'm Chiok Woo. I'll be presenting my study title, Morinda from Morinda citrifolia as a potential anti-proliferative agent against colorectal cancer cell lines. First, let me introduce you the plant, Morinda citrifolia, which is also known as noni fruit worldwide, or mengkudu in Malaysia. It can be consumed or used as traditional folk medicine. It is a small evergreen tree distributed in tropical climate zone, and it contains a wide diversity of phytochemical substances such as phenols, saccharides, flavonoids, and anthraquinones. So anthraquinone is our compound of interest. It is an aromatic organic compound with 9,10-anthraquinone, which can be found abundantly in roots and fruits of Morinda genus. Anthraquinone displays anti-inflammatory, antioxidant, anti-cancer, and antibacterial properties. Colorectal cancer is the third most frequent cancer worldwide and the second most common cancer in Malaysia. The risk factors of colorectal cancer include diet, alcohol intake, obesity, gender, family inheritance, and genetic. There are three types of colorectal cancer. First is inherited or hereditary. Second is familial. And third is sporadic colorectal cancer contribute about 60 to 70%. So sporadic colorectal cancer is a progressive accumulation of genetic alteration that lead to the transformation of novel epithelia to adenocarcinoma, which KRAS and P53 are among the most frequent mutated genes. Now, let's talk about p53 mutation in colorectal cancer. p53 is a tumor suppressor gene, and its mutation contributes to about 60% of the patients. The type of p53 mutation is a missense mutation and loss of wild-type p53, which leads to the excessive proliferation and unguarded entry in cell cycle. Studies suggested that autoregulatory negative feedback loop involving the regulation of p53 by MDM2 could be a novel target therapy in cancer treatment. So what is this loop? Under unstressed condition, p53 stimulates the expression of MDM2, while MDM2 in turn inhibits p53 by promoting its nuclear export for proteasomal degradation. While under stress condition, p53 still regulates MDM2, but MDM2 cannot inhibit p53, and this leads to the gene activation. KRAS and oncogene. So there are about 50% of KRAS mutation in colorectal cancer, especially in metastatic colorectal cancer. There is high mutation in codon 12 and 13 in the exon 2 of KRAS gene in colorectal cancer. KRAS mutation in colorectal cancer is always associated with a resistance to EGFR inhibitor treatment. Okay, so the problem statement for this study is there are less researchers focused on the anthraquinones from Morinda citrifolia as compared to anthraquinones from rhubarb and aloe vera. Second, many studies have been conducted on noni fruit juice, but little on the root part from Morinda citrifolia. Third, the effects of anthraquinones from Morinda citrifolia on MDM2 p53 interactions remains unclear. Fourth, to date, there are no effective drugs to deactivate mutated KRAS or selectively downregulating KRAS. Okay, so my methodology. There are 4 cell lines used in this study. 3 of them are colorectal cancer cells, which are HT29 cells with mutated p53, LS1740 with mutated KRAS, and HCT116 with mutated KRAS also, and 1 normal colon cell, which is a CTD841 colon normal. Here are the 8 anthraquinone compounds isolated from the root part of Morinda citrifolia. Okay. So from the first experiment that I performed is cell cytotoxicity test using MTT assay on 4 cells, 8 anthraquinone compounds, and 2 standard drugs. After screening, I proceed with 2 cell lines, which are HCT116 cells and HT29 cells, and 2 anthraquinone compounds, which are damnacantao and morindone. Then I proceed with cell proliferation test using Excel Legends real-time cell analyzer, gene expression analysis using RT-qPCR with primer targeting mutated KRAS, mutated p53, and wild-type MDM2. And then cell cycle analysis using flow cytometer, and finally whole transcriptome sequencing with morindone treatment only. Okay, here are my results. So this table shows the IC50 value obtained from MTT assay. So from these all these 8 anthraquinone compounds, damnacantao and morindone shows a potent cytotoxicity effect on 2 or more colorectal cancer cells. So from the IC50 value, we have calculated the selectivity index of the normal colon cells against colorectal cancer cells. And we can see that damnacantao and morindone show high selectivity index on colorectal cancer cells. Next, we have the real-time observation of cell proliferation of damnacantao and morindone treated HCT116 cells. For a better understanding, we shall see next slide. Okay, so from this bar chart, we can see that morindone exhibits higher inhibitory effect in HCT116 cells. And this is the real-time cell proliferation observation on HT29 cells. Okay, so from this bar chart, we can see that morindone demonstrated inhibitory effects at lower concentration and earlier than damnacantao in HT29 cells. Now we have qPCR results. This figure shows the gene expression of damnacantao and morindone treatment on mutated KRAS gene in HCT116 cells. we can see that the gene expression of damnacantao upregulates it, while morindone downregulates it by 1.9-fold change, which is what we are expecting. Then, as for HT29 cells with P53, mutated P53, damnacantao shows upregulation, while morindone shows downregulation by 2-fold change. As for wild-type MDM2 in HT29 cells, both damnacantao and morindone shows upregulation, but morindone treatment has a higher upregulation. In these slides, we have the cell cycle analysis results. And from the tabulated cell percentage, we observe a decrease of cell percentage in 24-hour, 48-hour, 72-hour in all 3 cell cycle phases in morindone-treated HCT16 cells as compared to the untreated. And this is the cell cycle analysis for HT29 cells. So in HT29 cells, in G2M phase, both damnacantao and morindone treated show lower cell percentage in G2M phase when compared to untreated. Next, we have the whole transcriptome sequencing data of HCT16 cells. So this is after performing differential gene expression analysis, we obtained the volcano plot and we obtained the heat map and the volcano plot of top 100 significant genes. And then we also obtained the top 10 most upregulated and most downregulated genes in HCT16 cells. And this is the KGG pathway analysis. Next, we have the gene ontology analysis of the top 10 most upregulated and top 10 most downregulated. And we shall do further investigation in all these components, in cellular component, molecular function, and biological process. And here are the heat map volcano plot in HT29 cells. And here's the gene ontology analysis. Okay, so for our 3 target genes, both whole transcriptome sequencing and RT-qPCR show similar results, which is downregulation for KRAS and P53 and upregulation for MDM2. Okay, so my discussion is damnacantao and morindone show significant cytotoxicity effect in both HCT16 and HT29 cells. Morindone demonstrates better anti-proliferative effects in both HCT16 and HT29 cells. Morindone downregulates the P53 and KRAS gene in HT29 and HCT16 cells respectively, while it promotes higher upregulation of the MDM2 gene in HT29 cells as compared to the damnacantao. The differential gene expression analysis on whole transcriptome sequencing also demonstrates similar results. A decrease of cell percentage across all 3 cell cycle phases in each time point was observed in morindone treated compared to untreated HCT16 cells. And both damnacantao and morindone treated HT29 cells show lower cell percentage in G2M phase when compared to untreated. So, as a conclusion, morindone may act as a competitive anti-proliferative agent in colorectal cancer cell lines. This research is supported by the grant IIRG 003C 2019 from University Malaya, and we have submitted a manuscript for this research. Okay, here are our references, and that's all from me. Thank you.
Thank you, Ms. Chi. We are now move on to our third presenter or representative, Mr. Muhammad Daniel bin Muhammad Effendi Kun from UiTM. Mr. Muhammad Daniel will be presenting his research finding entitled TLF Improve Gross Liver Morphology and Liver Histological Changes in Non-Alcoholic Fatty Liver Disease Mice Model. Are you ready, Daniel?
Ready, Doctor. Okay. Right, thank you, Professor Dr. Hamimah, for the kind introduction. I am Daniel Effendi Gun, and we are from University Technology Mara, Faculty of Medicine. And the title of our research is Tocotrienol Risk Fraction Improves Gross Liver Morphology and Liver Histological Changes in Non-Alcoholic Fatty Liver Disease Mice Model. So this is the content of my presentation. First and foremost, let me explain about what is non-alcoholic fatty liver disease. It's a spectrum of liver disease started with excessive fat infiltration into the liver due to high-fat diet. And the endpoint of this liver disease is non-alcoholic steatohepatitis, inflammation of the liver, and they deteriorate into cirrhosis and hepatocellular carcinoma. The global prevalence of NAFLD, the highest is in the Middle East country, followed by South America, and as Asian country stood at the third, 27.37%. For the past 10 to 15 years, the prevalence has been increased from 20% up to 27% and has projected to overcome the prevalence of hepatitis B and C. In Malaysia, the prevalence is about 35 to 50% and highest among type 2 diabetes patients. So the risk factor of NAFLD is of being obese, having metabolic syndrome and type 2 diabetes. And this patient mainly elder male, having high BMI, large waist circumference, and poor liver function test. And high fat deposition is fueled by the high fat diet, and this situation will induce oxidative stress due to lipid peroxidation. And apart from that, NAFLD condition also downregulates the nuclear receptor. The fascinal S receptor is a complex system that regulates and mobilizes and reuptake the bile acid. And downregulation of FSR will cause bile acid spillage into the blood circulation. And because of the oxidative stress due to lipid peroxidation, it drives the pathogenesis of NAFLD into non-alcoholic steatohepatitis and cirrhosis and hepatocellular carcinoma. Among the gold standard of treatment for non-alcoholic fatty liver disease is exercise, weight loss, and dietary modification. It's a patient-centric, but often generate poor compliance among patients. And other pharmacological treatment include anti-hyperglycemic, such as metformin, lipid-lowering agents, statin, nuclear receptor agonists, and also antioxidant. But this also comes with adverse effect, and especially antioxidants often produce inconsistent in terms of their outcome in NAFLD patient. So the scope of our research is the use of pump-based tocotrienol risk fraction. And tocotrienol risk fraction consists of two main isoforms, the tocopherol and tocotrienol, and each isoform consists of alpha, beta, gamma, and delta. And previous studies have shown, in general, that tocotrienol risk fraction able to elicit antioxidant activities by scavenging radical oxygen species, decrease pro-inflammatory mediators such as IL-6 and TNF-alpha, and also improve hepatic endoplasmic reticulum stress. This is very important, especially in NAFLD condition. Previous studies, if we look at NAFLD itself, there are a few studies that have been conducted in NAFLD, but this patient usually has been progressed to NASH. So they have been using only isoforms such as alpha tocopherol, gamma tocotrienol. And in fact, a study using TRF supplementation, but it doesn't show the biochemical or at genetic level the changes, but only improvement of hepatic echogenic response. So studies have been conducted mainly focused on non-alcoholic steatohepatitis when it comes to the use of tocotrienol risk fraction. So there's a lot of lack in studying on NAFLD condition, because this is very important to study on the earlier part rather than the deteriorate part. And it's also imperative to study in Malaysia because the prevalence is being increasing drastically for the past 10 years. And previous studies has been using vitamin E, because this is like an anecdote where they claim they're using vitamin E, but actually they're using specific tocopherol isoforms instead. So it's very important that we elucidate the role of pump-based tocotrienol risk fraction in non-alcoholic fatty liver disease. So the research of our objective is to first see what is the effect of TRF on anthropometric data in animal model, such as mouse body weight, food intake, random blood glucose, abdominal circumference. We look at the antioxidant activity, especially as superoxide dismutase and glutathione. And also we observe what the histological changes in the liver, and also we'll see what is the expression level of farnesyl S receptor through immunohistochemistry staining. So our research design consists of the use of genetically modified dietary induced NAFLD, where we use the leptin knockout mice shown here. they'll be producing obesity and also NAFLD once induced with 60% high-fat diet. So these 28 mice were randomly divided into 4 groups, where the positive control consists of high-fat diet only. Then the second group, high-fat diet with palm kernel oil. The palm kernel oil is the carrier for our TRF, especially the enhanced topotrienol risk fraction. So the third group consists of high-fat diet with TRF, and the fourth is the enhanced one, ETRF, whereby TRF consists of 70% tocotrienol and 30% tocopherol, and combined with palm oil, which mainly consists of long-chain triglyceride. And ETRF is 75% tocotrienol, 35% tocopherol, and palm kernel oil as its carrier, medium-chain triglyceride. The difference here is that the MCT has shown through previous study that it's able to bypass the circulation and being directed straight into the portal circulation into the liver, increasing the bioavailability of TRF. So these mice were given treatment for 6 weeks, whereby ETRF, ATRF, and also PKO were given at 200 milligram per kilogram every day. Throughout each week, we measure the body weight and food intake. Towards the end of the study, abdominal circumference and random blood glucose were measured before being sacrificed, and the liver were harvested for antioxidant activity measurement, histological analysis, IC analysis as well. So the TRF supplementation shows that able to affect the anthropometric indices and dietary intake, but no outcome was observed on random blood glucose in NAFLD model. So TRF able to produce the lowest body weight compared to other groups, although it able to take the highest food intake compared to other groups. So here we can see that TRF able to induce highest body weight to a food intake ratio, meaning that it able to eat a lot, but able to maintain its weight compared to other group. But it unable to control its abdominal circumference, random blood glucose, and antioxidant activities. In mice that is induced with NAFLD, is shown with statotic liver, whereby we can see a whole range of statosis yellow patches, and most of the liver are swollen. But with the TRF and ATRF treatment, we can see that there's only patches of statosis on the surface of the liver. Same goes to palm kernel oil. So here we can see that there's a less severe statosis in the liver when these mice were given TRF and ATRF supplementation. But the most important thing is that the liver span of the TRF JAX mice was smaller compared to high-fat diet and TRF. So in terms of histological analysis, the high-fat diet only mice show classical presentation in the liver of statotic liver, whereby it consists of macrovascular statosis at high magnification and also presence of neutrophils indicating inflammation. So there is a pan-acina statosis consisting of zone 1 and to zone 3. But in TRF and ATRF supplemented mice, they only show not pan-acina, but certain region of the statosis in ETRF, but not in TRF. I will show later based on our quantitative analysis. In terms of Mason tractome staining, to see any fibrotic changes, we can see there's a fibrosis in high-fat diet mice, but not in ETRF, but a little in TRF-treated mice. So the histological analysis is being quantified based on NAFLD activity score to show what is the risk of developing cirrhosis and NASH. So we can see here that the ETRF-treated mice has lower NAS compared to other group, whereby ETRF supplemented mice although have high grade of statosis, they're only localized to only zone 3, no fibrosis, and very low lobular inflammation compared to TRF having mid-grade, but the statosis encompasses of the whole region of the liver and has high lobular inflammation. So it's very important to see that the higher bioavailability of the ETRF able to control the statosis of the liver. In terms of FXR expression, we can see that ETRF has higher FXR expression compared to other groups by looking at the chromogen expression inside the cell of the hepatocytes at the nuclear level. But I will show in terms of the quantitative analysis. So here shows that the ETRF has higher FSR expression based on the positive cell intensity of the staining compared to other groups. So what we observe is due to the most likely to the mechanism of the MCT itself able to increase the bioavailability of TRF inside the liver. Previous study also have shown that the combination with MCT have able to increase, and with the increase in bioavailability, able to increase the beta oxidation and reduce the radical oxygen species. This is also supported by our untargeted metabolomics profiling in these mice. And increase in FSI expression also been observed in ETRF treated mice. Therefore, we can see that there's only zone 3 macro and microsteatosis with smaller liver span and decrease in lobular inflammation foci, and also eventually will decrease the risk of having NASH and also cirrhosis and hepatocellular carcinoma. Whereas in TRF treated and only high-fat diet consistent in having pan-acina macro and microsteatosis. So as a conclusion, TRF only able to address the anthropometric and biological outcome, because with the combination of LCT, it follows the peripheral circulation. It will only able to limit the peripheral adipose tissue expansion, but not in the liver. That's why we can see that there's an effect in controlling the weight. Whereas ETRF addresses the concern in NAFLD, whereby able to limit the progress of steatosis in the liver, where we see that there's only zone 3 steatosis, and this is contributed mainly by the MCT in increasing the viability of ETRF. With that, this is the reference of my presentation. I would like to acknowledge UITM, ICMBB, Animal Lab, and also Faculty of Anatomy Laboratory, and also SAMDABI all in contributing the ETRF and ETRF. And this is a list of our grant. Thank you.
Thank you, Mr. Daniel.
Welcome, Doctor.
Now we move on to our next presenter, oral presenter, Miss Anna Andrew from USM, with a presentation title of Generation of DNA Aptamer Against Envelope 2, Protein of Chikungunya Virus by In Vitro Systematic Evaluation of Ligands for Exponential Enrichment for Diagnostic Applications. Yes, Miss Anna, you may begin now.
Okay, thank you, Dr. Siti Hamimah. So, good morning, everyone. My name is Anna. So today I'm going to present research entitled Generation of DNA Aptamers Against Envelope 2 E2 Protein of Chikungunya Virus by In Vitro Selects for Diagnostic Application. So, chikungunya can be referred to the virus as well as the disease. So, chikungunya virus causes chikungunya disease or chikungunya infection. And the name chikungunya is from Makonde language, which means to walk bend down, a posture adapted by patient affected by chikungunya due to extra joint pain. Okay, and chikungunya is a mosquito viral disease where Aedes aegypti and Aedes albopictus are the two mosquitoes involved in human transmission. So envelope 2, E2 protein was chosen in this study. Why E2 protein? Because E2 forms spikes on the virus surface. Because it is prominently exposed on the virus surface, it can be easily detected. And this is good for diagnostic application. And during viral infection, E2 also involved in receptor binding and allows cell entry. So antibody elicited by immune system recognize epitope and E2. Therefore, E2 serves as a good serology detection marker for chikungunya virus. And there are many different diagnostic tests available for chikungunya virus, and the choice of tests depends merely on the duration of patient symptoms. So the symptom generally lasts for 5 days in which virus is present in the patient sample. So we can use viral isolation and molecular technique to detect the virus as well as the viral genome. And we can also use immunoassay technique to detect the antigen. Okay, and then the antibodies against chikungunya can be detected 5 to 10 days after post-illness onset, and the antibody can be detected using immunoassay technique. And among all of these tests, virus isolation and molecular technique offer the most sensitive and specific method. However, these two tests require specialized laboratory and then expensive equipment and reagent. The most cost-effective and user-friendly will be using immunoassay technique, especially in resource-limited laboratory. So what is the problem with the current immunoassay? This immunoassay use antibody, and the antibody-based immunoassay, they are prone to batch variations, and then they are very expensive and they are less stable in harsh temperature. And currently, there's no widely available chick virus commercial assay, especially antigen detection tests. Antigen detection tests will be a good alternative to virus isolation and molecular technique to detect or to diagnose a patient in the early phase of infection. So when there is no laboratory test for chikungunya, it will cause delay in diagnosis or undiagnosed chick virus patient, and this will lead to unnecessary use of antimicrobial, where this is not only a waste of resources, but it will also increase the risk of antimicrobial resistance. And when there is delay in diagnosis, it will also cause lab intervention such as vector control strategy, and this will also cause further spread of the disease. So aptamers, they are a good alternatives to antibody. So what are actually aptamers? They are single-stranded nucleic acid, DNA or RNA that has capability to fall into three-dimensional structure, and this will allow them to interact with the target. So they are also known as chemical antibodies because just like antibodies, they bind to the target via induced fit mechanism. So aptamers serve as a good molecular recognition elements for the development of diagnostic tests because aptamers, by their very nature, they are sequence defined, they are chemically synthesized. Therefore, they are very stable, they are cost effective, and they are highly consistent, and they can be easily modified as compared to antibodies. So the objective of the study was to generate DNA aptamers against E2 chikungunya virus and to characterize these aptamers. So for methodology, first we clone, express, and purify the chick virus E2 protein. And this E2 protein were used to generate DNA aptamers by selects. And finally, we characterize these DNA aptamers. So cloning and expression of the E2 gene were done using bacterial expression system. So after we have the protein, we purify the protein using column chromatography method, and we further verify the protein using Western blot analysis by using different antibodies. And then the purified protein were used to generate DNA aptamers by selects. So in one select cycle, it involves three main steps. The first one is selection, second partitioning, and third one is amplification. So in this study, what we did is the purified protein we incubate with a pool of single-stranded DNA library. And this single-stranded DNA library consists of 60 randomized internal sequence flanked by 2 constant regions. And then after incubation, then we will remove those unbound nucleic acid. And we did it using dual partitioning method. First, we use a nitrocellulose membrane. Second, we use microtiter plate. And finally, we amplify the bound nucleic acid using a normal PCR protocol. And before this nucleic acid pool is used in the next cycle, the double-stranded DNA is converted into single-stranded DNA using lambda exonuclease digestion. So the select cycle is repeated until functional molecule dominate the population, at which there will be clone and sequence. So after we got the sequence, we will have a DNA aptamer candidates, and we will characterize this DNA candidate first by looking at its secondary structure using M4 web server, and then we will determine the binding affinity of the aptamers by using ELISA method. So binding affinity is defined as the strength of interaction between aptamers and the protein. So what we did here is we use a serial dilution of the protein coated on the plate, and we incubate with a fixed amount of biotinylated aptamers and detect the binding affinity using a calculation using a GraphPad Prism software. So, and then we develop a direct antigen detection test for chikungunya virus using the aptamers. So what we did here is we spiked the recombinant protein into a healthy serum, and we coated it on a plate, and we incubate with biotinylated aptamer, and then we determine the limit of detection. So for result and discussion, so we have successfully purified the protein. This is purified protein, and then we dialyze the protein, and the dialyzed protein will further analyze with antibodies. So the first one is using nickel antibody, which will detect the 6-histidine in the recombinant protein, and then we detect with chick positive serum and anti-chikungunya polyclonal antibody. And there is no cross-reactivity when we tested it with dengue serum. And then after 9 select cycle, we send the nucleic acid pool for sequencing, and the sequence revealed there are 6 potential DNA candidate, with CHIK2 has the highest frequency, 61.9%. So we further characterize this CHIK2 aptamer, and this is the predicted secondary structure of CHIK2 aptamer, which have 2 stem loop structure, one big and one small loop, and stem loop structure play a fundamental role in target molecule binding. And the binding affinity of these aptamers are 177.5 nanomolar, which is comparable to monoclonal antibody that usually range from nanomolar to picomolar. And the preliminary result for diagnostic application using this direct ELISA showed that the limit of detections is 52 nanomolar. And so far, there was only one study who have generated DNA aptamers against chikungunya virus, and they used E1A peptide as a target. And they characterize this aptamer only based on the ELISA absorbance. The higher the absorbance, the better the aptamers. And then they use two different aptamers to develop a lateral flow assay. And they have shown that this lateral flow assay can be used to detect the E1A peptide as well as the envelope protein of chikungunya virus. So as a conclusion, we have purified the recombinant E2 protein, and this E2 protein we use as a target to generate DNA aptamers. And after 9 select cycle, we have 6 potential DNA aptamer candidates, and we have only characterized one of it, which have KD of 177.5 nanomolar. And we have also shown that CHIK2 aptamer has great potential for development of diagnostic system for chikungunya virus. And we hope that this aptamer-based immunoassay can be used to develop a diagnostic test that will meet a sure characteristic recommended by WHO. So with that, I would like to thank my supervisor, Prof. Tang, my co-supervisor, Dr. Chita Tan, and Dr. Magdalene, and all of my colleagues and friends in RNA Bio Group. Thank you.
Thank you, Ms. Anna. Yes. Now, we are at the end of the session with our last oral presenter, Mr. Mizano Fakir Ghazali from UiTM. He will be presenting his research finding titled Expression of Specific Biomarker towards Microfold Cells Differentiation in 3D Culture of CACO2 and IgB cells. Are you ready, Mr. Mizano?
Yes, Doctor. Can you see my
you
slide?
may begin now.
Is my slide visible?
Yes, I can see your slide.
So yeah, okay. Thank you, Madam Chairperson, Professor Dr. Sita Mimah. Assalamualaikum, very good morning to all participants and also all the audience. My name is Mizano Fakri from Faculty of Medicine, University Technology Mara, Malaysia. And today I would like to present the expression of specific biomarkers toward microfold cells differentiation in 3D culture of CACO2 and IgB cells. So this presentation will cover the background of study, the research question, objective of study, materials and methods, results, and conclusion. So microfold cells, also known as the M cells, is a specialized cells found in the follicle-associated epithelial of the patches in human intestine. So it have a unique morphology, which are the irregular brush border, reduced glycocalyx, and sparse microvilli. So this morphology allows transcytosis, which is the movement of small molecule across the intestinal barrier. However, this morphology also allows pathogen to manipulate and avoid the intestinal barrier prior to infection. Given that the specific markers for M cells, such as CA99, alpha-5 beta-1 integrin, Ulex serapes antigen with germ agglutinin, and also glycoprotein 2, which may assist the pathogen during the infection. So in this study applied 3-dimensional cell culture system as compared to the common 2-dimensional cell culture system. Basically, 2D cell culture system is a system where the cells adhere and grow on an artificial flat surface, such as tissue culture flask. Meanwhile, for 3D cell culture system is a system where the cells suspended in an artificial matrix, which mimics the environment of the cell in vivo better. Apart from that, the shape of 2D cells are mostly flat and stretched, while for 3D cell culture system, it produces spheroid-shaped cells with the presence of extracellular matrix. So the question that arises in this study is that how is the M cell differentiate in 3D culture models, and is M cell differentiation able to induce the expression of its protein markers? Therefore, the objective of this study is to investigate the expression of M cell markers in 3D culture models and the specific objectives include to determine the level of differentiation of KECO2 cells into M cells induced by Raji-B cells via transepithelial electric resistance tear measurement, and to measure the expression of specific M cells markers using Western blot analysis. So this study implied two types of 3D culture system. The first one is the 3D co-culture system using transmembrane wave plate with 3 micrometer porous membrane. Briefly, KECO2 cells was seeded into the upper compartment of membrane insert which represent the apical site of human intestine. The cells were maintained for 14 days and the tear value was measured daily. Well, tear measurement is a test to measure the resistance of electrics to pass through the cell monolayer which indicate the integrity and polarity of monolayer formation. So for the second 3D culture system, the second 3D culture system used was arginate hydrogel beads. Briefly, KECO2 cells was suspended in arginate sodium chloride solution. The suspension was then dropped into a petri dish containing calcium chloride using 25 gauge needle syringe. The cells were maintained for 14 days before Raji-B cells was introduced into the lower compartment of membrane insert and onto the petri dish. So the cultures were then further incubated for another 7 days to induce the formation of M cells. Finally, total protein was extracted, quantitated, and undergo Western blot to determine the expression of CA99, alpha-5, and beta-1. So move on to the results. KECO2 cells appeared as polygonal polarized adherent cells, while Raji-B cells presented as lymphoblast-like suspension cells. For the monoculture on transmembrane, KECO2 cells monoculture on day 3 showed lack of cell formation with clear area was observed. Meanwhile, on day 14, KECO2 cells monoculture showed a compact and tight formation. For the monoculture in arginate hydrogel beads, KECO2 cells monoculture on day 0 showed dispersion of single cells, while on day 14, KECO2 cells monoculture appeared packed and dense. This histogram showed the value of the tear values recorded from day 0 until day 21. The final tear values were calculated using given formula, and it was observed that the resistance values started to increase from day 2 until day 14 of incubation. After Raji-B cells was introduced on day 14, the resistance value started to decrease and consistently reached below 50 ohm centimeter square on the subsequent day until day 21. So the expression of CA99, alpha-5, and beta-1 by C, KECO2 monoculture, and both co-culture models of T, transmembrane system, and B, arginate hydrogel bead system, were detected via Western blot. It was observed that there was a single band at approximately 140 to 150 kilodalton for beta-1, and two bands of full length and light chain of alpha-5 for 3D transmembrane system was observed on all samples. In addition, the expression of alpha-5 was upregulated in 3D transmembrane system as compared to the KECO2 cell monoculture. So this study was in parallel and supported the previous study that has been reported. Meanwhile, no detection was observed for CA99. So in this study, the tear value was expected to reach between 150 to 400 ohm centimeter square, which indicates that the monolayer is fully differentiated and ready to be induced by Raji-B cells. At post 14 days of incubation, the tear value was 244.8 ohm centimeter square within the range, and the value drastically dropped after introduction of Raji-B cells. So this finding justified that the monolayer integrity was reduced, probably due to the disorganization of cells tight junction with the presence of RGB cells which compromise the monolayer structure. As for Western blot analysis, all the specific markers were expected to be upregulated. However, in this study, no detection of CA99, which may due to non-compatible antibody that specifically detects CA99 expressed by KECO2 or any other intestinal cells was observed. Apart from that, for both alpha-5 and beta-1, previous study reported that the heterodimer of alpha-5 and beta-1 integrin complex were upregulated in M cells. Based on this study, the upregulation might be due to the expression of alpha-5 subunit only. So to conclude, both types of 3D culture system were supportive towards the M cell differentiation indicated by presence of alpha-5 and beta-1 integrins, although at different level of expression. Thus, both integrin proteins are suitable as markers for M cells. So these are the references that I referred to for the study. And I would like to acknowledge Ministry of Education Malaysia for FRCS grant, Institute of Medical and Molecular Biotechnology, ICMBB, Faculty of Medicine, Biotechnology Mara, Malaysia, and also Synthetic Biology, Engineering and Regeneration, Cyber Group. With that, I thank you.
Thank you, Mr. Mizano. With that, we end our oral presentations. Now we move on to the Q&A sessions. The first question is for Prof. Effendi. Are you here, Prof. Effendi?
Yes.
Yes. The first question in the chat box from Dr. Amira. The question is, how often do you have to apply the chitosan hydrogel on the wounds? And apart from tilapia fish, can chitin from other fish be used, or are they less superior compared to the tilapia?
Thank you, Dr. Siti Amimah. Thank you for Siti Amira. So we apply chitosan hydrogel twice a day, actually, on topical application, direct on the wound. And based on the literature review search, can you hear me?
Yes, Prof.
Can you hear me?
Yes, Prof. I can hear you, Prof.
Okay, based on the literature search, 99% of fish scales have very similar profiles, which made out of 97% chitin. So we are converting the chitin. So basically, any scales should produce very similar results. So it's okay if you want to choose another fish, because we have fish with big scales like tempurau and also arowana, but they are high valued fish. That's why we didn't choose them, because they have another values in the aquaculture industry. And like I said before, tilapia is a very popular fish, so they are produced in mass volume. You can also use krills or shrimps because they produce 40% chitin compared to fish, 32% of chitin. But in fish, the volumes are greater than krills and also shrimps. That's why we choose fish scales. And bear in mind that there are thick and thin scales of fish. So we are using tilapia because tilapia scales are just nice, and they are readily available in commercial market, actually.
Next question, Prof. There's another question from Mr. Azizat. The first question from him is, did the scale crush before chitosan extraction? The first one. The second one, how often the chitosan treatment is applied on the wound in the 21st day of period? Thirdly, why only male mice is used in your study? Prof, do you mind to turn on your camera?
Yeah.
Thank you, Prof.
Okay, we don't need to crush the scales before chitosan extraction. This is because it gets dissolved in acid digestion. During the deproteinization, we treat it with 8 molar hydrochloric acid under pressure for 12 minutes in the autoclave. So this will dissolve the glucosamine group and will form the residue on the bottom of the flask. So we collect the residue and proceed for deacidification process. We want to avoid any hormonal effects if we use females white rat, because as you know, they are always in hormonal imbalance if they are matured. Let's say for female white rats, they also sometimes goes into estrous cycles. Their estrous cycles might also affect the healing process. So that's why we use all males, because males are not really affected by hormonal changes, actually.
All right. Thank you, Prof. Effendi. Next question is for our second speaker, James. The first question is, James, on how long does it take you to have this analysis done? The first question. Is there a potential to use this to predict which patient is going to develop severe disease? Yes, so those are the two questions. And then do you want me to read the other question, or you want to answer the question?
One at a time, please. Yeah.
All right, sure. Go ahead.
Yeah. Well, yeah. So essentially, this particular process. I mean, so actually, just each individual experiment takes approximately 2 days. It's kind of a split overnight thing because we use a kind of a overnight formaldehyde kind of fixation, and it's not super fast, and also because the data analysis itself is relatively time consuming. So it's something we are considering of can we like do this fast enough to make it clinically useful? So for example, taking a sample, and actually because we do know, for example, in our current data sets, so there are some patients who actually changed their designation like from the point of taking blood and then later on. So we had one sample which the blood sample was taken at the point they were considered a moderate case, essentially, and that was when the blood was taken. But later on, they became critical. And actually, when we analyzed that blood sample, we found they were very much phenotypically in terms of their cellular populations in the critical group. So in that case, we weren't able to do that fast enough to be beneficial to that particular patient. But it was absolutely the case that, while clinically they were moderate at the point of giving blood, their immune populations were already of a critical kind of type, so actually it was only one patient, so we can't say too much about it. But there was some hint that actually, we could predict things from that. But we haven't done that so much so far.
All right. The next question is if a person receives a vaccine, do you expect the profile would be similar to the COVID-19 patient, or the profile would be different?
So we expect, kind of pretty different, I guess, because of course, the actual infection is a very severe infection. There's a lot going on in the immune system, whereas vaccination, depending on a particular type of vaccine, is obviously a much more targeted approach. And we are actually collecting samples from vaccinated individuals, and we will be looking at kind of cellular changes. But I think the prediction would be there may be some similarities, which would be very interesting, but we don't expect to see this kind of level of broad activation in vaccinated individuals, because, that wouldn't be a good thing if that was happening, because as you can imagine, vaccines shouldn't be inducing, severe immune kind of phenotype changes. So it should be much more specific to antigen specific. So we'll be looking at plasma cell TFH and such like, but we don't expect such aggressive broad changes.
What about your view in terms of, would there be any differences when patients receive different, or people receive different vaccine, be it like Pfizer, AZ, or Sinovac? Do you have any view on this? Have you ever come across any study that actually reporting this?
No, so I think it's not that well characterized at this time. We probably do expect some differences. So for example, the Pfizer vaccine, it's using an mRNA, and actually the mRNA is both the, so of course it's there to kind of produce the protein, but it's also as the adjuvant itself. So it's going through kind of TLR7 signaling into the innate immune system. And actually you need that mRNA adjuvant effect to get the good vaccine effect, because they also demonstrated in other work. But if you actually modify the mRNA to remove its adjuvant effect, it becomes a kind of a tolerogenic vaccine. And they're using, so BioNTech are kind of like trying to work that through for autoimmune vaccination. So I suspect, the details will vary between vaccines, but I mean, I guess in terms of the end stage populations they're targeting, so of course we expect in all of them to be generating, antigen-specific antibody responses. And then of course the other question is to what extent, are they signaling initially through the innate immune system, and to what extent you're getting kind of good development of T-cell immunity as well is maybe not so well characterized at this time, because we tend to use antibody response as a marker of good vaccine responses. But sometimes, the level of involvement of the T-cell, the various T-cell subpopulations, T follicular helper being an obvious one, isn't so well characterized at this time. And it may vary between different vaccines.
Thank you, Prof. Effendi and Dr. Wings, for extensive answer from all the participants' questions. Thank you so much. And then we move on to our first oral presenter. There are actually quite a number of questions. Can I remind all oral presenters to switch on their camera? The first question is to Ms. Chi. Did you synchronize the cell before compound treatments? Second question, did you check the gene affected during cell cycle arrest? Yeah, so 2 questions. The third question is, did you validate by Western blot for KRAS and the other targeted genes?
Thank you for the questions. So for the first question, I did not synchronize the cells because I want to see the effects in all the phases of cell cycle after the treatment. And for question 2, from our cell cycle data, there's no pronounced cell cycle arrest, but rather cell cycle accumulation in the phases. So it is not really essential for this study so far to look at the genes that cause cell cycle. And for third question, yes, we will validate this with a Western blot later on. Thank you.
All right, thank you, Ms. G. There's one question for Mr. Daniel. The question is, may I know how do you choose the treatment period of 6 weeks? And was the FXR analyzed by Western blot?
Okay, thank you, Dr. Amima. And thank you for the question. So we decided 6 weeks after reviewing extensively in terms of the animal model and the treatment that given to this animal model. And the animal model that we're using is, I would say, highly accelerated in terms of developing the NAFL because they are leptinemia. So they have this unsatiated appetite towards the high-fat diet. So within the period of acclimatization, in fact, they already developed the NAFL. So that's why that we seem the best and ideal period for supplementation is between 4 to 8 weeks. So we take it 6 weeks. It's the most ideal timeline to develop ideal result. And also able to, because we also do untargeted metabolomics. So within 6 weeks, also able to derive all the metabolites to be picked up by the analysis. And in terms of the FXR expression, we do it through immunohistochemistry staining instead of Western blot because it's very important to see the spatial localization of the FSR expression when it gives under supplementation of TRF, ATRF, because we want to see whether the FSR expression is localized between zone 3, 201 or the other way around, or there's a specific region that controls the expression of the fatty acid beta oxidation. And we compare them with the HNA staining. So rather than doing Western blot, we go for immunohistochemistry staining. And we derive the result in terms of quantitative analysis through scoring. Thank you.
All right, thank you, Daniel. The next question is for Mr. Mizano. The first question is, how do you harvest the cell from the alginate bits?
Okay, so thanks for the lovely question. So basically, in this study, the alginate bits can be disintegrated and can be solubilized by using a sodium citrate solution. So when the disintegration was performed, later on, just do the certification to recover the cell. And then for the second question is all alginate bits regular in their size. Well, approximately the size of each alginate bit was approximately 10 millimeter cubic. 10 millimeter cubic. So the size is approximately the same because the distance of the solution drop into the petri dish is standardized. So I hope that answered the question.
Yes. Thank you, Mr. Mizano. Any other question from the participant, from the audience? If there's no more question, can I ask all the participants and audience to turn off the camera so we can take a picture together?
Okay, sure.
Is everybody ready? 1, 2, 3. Just a few pages actually. Thank you, everybody. Thank you for your time. And I just want to remind you to join our next session at 11:20. Should I pass it back to Dr. Wong Sok Moy for any announcement?
Sorry, I have a phone call. Okay, sorry. Okay. I don't know how to switch off the phone. Anyway, we will have a short break, okay, and come back at 11:10, right, Dr. Bima? Okay, see you then. Okay.
Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture. Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture. Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture. Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture. Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture. Puzzled by your sequencing results? You've seen the incomplete reads. You've seen the inaccurate of short reads and long reads to help you unlock the secrets of even the most complex genomes. Sound too good to be true? Go here to get the whole picture. Puzzled by your sequencing results? You've seen the incomplete picture of short reads. You've seen the inaccuracies of traditional long reads. PacBio Hi-Fi sequencing gives you the whole picture. Hi-Fi reads combine the best of short reads and long reads to help you unlock the secrets of even the most complex genomes.
Salam sejahtera and very good morning to all participants. Welcome to the prize presentations and closing ceremony of the 4th International Conference on Molecular Biology and Biotechnology. My name is Siok Mui, and I will be the MC for this session. We will now move to the final part of the conference, the awards ceremony. I'm sure every one of you, especially the presenters, have been waiting anxiously for this moment. Before we announce the results for Best Poster Presentation, I would like to announce several prestigious awards awarded by the Malaysian Society for Molecular Biology and Biotechnology to MSMBB members. MSMBB EduSTEM Grant. MSMBB EduSTEM Grant was set up in 2019 to support projects working with science, technology, engineering, and mathematics education, and to fund activities which are aligned with UNESCO Sustainable Development Goals, such as to improve the quality of education, life, health, and well-being. The winner of MSMBB EduSTEM grants goes to Associate Professor Dr. Siti Sarah Othman with the project title, Are You Human or AI? See Your DNA. And Dr. Adeline Song Ai Lian with the project title, Evoking an Interest in STEM Through the Mysterious World of the Unseen. Congratulations to Dr. Sarah and Dr. Adeline. Next is the MSMBB EduSTEM X grant. This grant was set up in 2020 as a seed fund to support projects working with virtual STEM education. Preference is given to projects which demonstrate innovative approaches and techniques to solve community issues and build individual, neighborhood, and community capacity. The project must bring direct benefit to the community. The winner of MSMBB EduSTEM X grant goes to Dr. Kyo Sina with project title, Enhancing Students' STEM Knowledge and Language Proficiency through Flipped Classroom Method in Online Learning Platform. And Associate Professor Dr. Crystal Lim Siu Yin with project title, STEM Food Fun, Learning the Art and Science through Science of Food, Casting the Science Classroom through an inquiry-based pedagogy. Congratulations, Dr. Kyo and Dr. Crystal. Next, we move on to MSMBB Best Scientist Awards. This year, MSMBB has established the Best Scientist Awards to recognize outstanding scientists who have made significant contributions to the field of molecular biology and biotechnology. There are two categories of awards, namely Distinguished Scientist Award for members who are above 40 years old and Young Scientist Award for members who are at the age of 40 or below. The winners of MSMBB Distinguished Scientist Awards goes to Professor Dr. Ko Chong Lek. Prof. Ko, can you please turn on your video, please? Are you around? Is Prof. Ko here? All right. Okay. I would like to read out a brief biography of Prof. Kaur. Dr. Kaur is an acknowledged doyen of genetics, molecular biology, and biotechnology in Malaysia and the Asia-Pacific region. Since 1981, he has passionately pioneered, championed, and provided dynamic leadership in the advancement of molecular biology and biotechnology in Malaysia. He was featured in many Malaysian newspapers and magazines, some of which referred to him as the DNA man, Bapak DNA, and the Genetics Man. A special Momentum 2 documentary series, episode 19, The Blueprint of Life by Prof. Kaur Chong Lek, was filmed and screened on 13 December 2001 on Astro Ria Channel. From July 1970 to June 2018, he had 48 years of eminent academic record at universities and research institutes in Malaysia, Singapore, England, Germany, Japan, and the US. From July 2018, he is the Chief Scientific Officer of Gene Express Sdn Bhd, a direct-to-consumer DNA testing company. Dr. Kaur and his research associates had used molecular biology and genetic techniques to investigate the genetic materials of various organisms. In 1993, he was the first scientist in Malaysia to provide DNA typing service for human identification and parentage testing. In 2000, he was a member of the team that discovered Malaysian island flying foxes as the natural animal reservoir of Nipah virus. He holds a BSc Honours and MSc in Genetics from the University of Malaya and a PhD in Biochemistry and DIC from the University of London and Imperial College of Science and Technology, England, respectively. He is a Fellow of the Academy of Sciences Malaysia and a founder member of the Asia-Pacific International Molecular Biology Network. He has received many awards and fellowships, including the 2001 ICMBB Applied Biosystem Award and the Malaysia Torrey Science Foundation's 1996 Science and Torrey Technology Award. He has used his expertise to serve the academia, community, government, and industry. Currently, he is actively promoting biotechnopreneurship. Congratulations, Prof. Kaur. Okay, is Prof. Kaur around? All right, then it's okay. We'll move to the next Best Scientist Award. Is the Young Scientist Award. The winner of MSMBB Young Scientist Award goes to Associate Professor Dr. Lionel In Lian Ong. Dr. Lionel, can you please turn on your video, please? Then Lionel, Dr. Lionel, are you around?
Yes, I'm here.
Yeah,
thank you.
Okay, congratulations. All right, okay. I will now like to read out a brief biography of Dr. Lionel. Okay. Associate Prof. Dr. Lionel holds a BSc in Biotechnology. Oh, sorry, I have to move. Okay. Associate Prof. Dr. Lionel holds a BSc in Biotechnology from UPM, a MSc in Biotechnology from the Malaysian University of Science and Technology, and a PhD in Molecular Oncology from University of Malaya. Dr. Lionel has been active advocate of research excellence and the advancement of science, technology, and innovation in Malaysia. He specializes in the area of cancer immunotherapeutics, and in 2015, he established the Cancer Immunotherapy Research Group at UCSI University. His current research involves the development of cancer vaccines, antibody engineering technologies, and cancer diagnostic. In 2017, he was awarded the US Innocentive Award for a new method describing the delivery of biomolecules through the gastrointestinal tract. He has also successfully secured several government and industry research grants amounting to RM 1.4 million, and currently holds 2 patents involving cancer therapeutics and vaccines. In 2019, Dr. Lionel was appointed as the Dean for the Faculty of Applied Sciences at UCSI University, where he spearheaded the establishment of various academic programs, including the Forensic Science and Applied Microbiology program. Dr. Lionel was also instrumental in initiating the faculty's International Research Attachment Program for student placement at top-ranked universities, such as Harvard and Yale University. He is also the chairman of the Institutional Ethics Committee, a KPT-appointed MIRA auditor, and head of research program for STI on environment and sustainability under the Tan Sri Omar Center for STI Policy. He also served 2 terms as the ICMBB Honorary Treasurer and is also a handling editor of the Asia Pacific Journal of Molecular Biology and Biotechnology. He is also a member of the Young Scientist Network under the Academy of Sciences Malaysia. He is engaged in many of its activities, including the Science Integrity Working Group and the Science in Excel initiative in collaboration with the World Academy of Sciences. All right. Thank you, Lionel. Would you like to say something?
I'm very honored for this award, and I hope this will inspire many young scientists to follow suit in the coming years. So thank you very much, MSMBB, for this wonderful honor. Thanks.
Thank you, Dr. Lionel. Again, congratulations to Prof. Koh Chong Lek and Associate Professor Dr. Lionel. All right. Finally, here come the highlight of the day, announcing the results of Best Presenter for 3 minutes poster presentation. We had 107 participants for our 3 MPP, which includes 11 undergraduate, 62 postgraduate, and 34 from the professional and industry, 78 under biotechnology, and 29 under molecular biology. The Best Poster Award will be awarded to the undergraduate, postgraduate, and professional presenters for category biotechnology and category molecular biology. All right. There is only one winner for the category biotechnology undergraduate. Best Poster Presenter for the category biotechnology undergraduate goes to Poster 111 Miss Annie Mei Ting Lau from Utah, with title Isolation and Characterization of Probiotic Bacteria from Livestock and Kimchi and Assessment of their Antibacterial Potentials. Congratulations, Miss Annie. All right, next. Okay, there are 5 winners for category biotechnology postgraduate, okay? And Best Poster Presenters for category biotechnology postgraduate goes to Poster 260, Miss Yu Xuan Hao from UCSI. Title, Optimization of Milk Fermentation Incorporated with Recombinant Lactococcus lactis NZ3900. Congratulations, Miss Yu. Next is Poster 241, Mr. Jun Bing Ng from UM. Title, Differential Lipocalin-1 Levels in Keratoconus in a Malaysia Cohort. Congratulations, Mr. Jun. Next is Poster 218, Ms. Ho Ping Chong from UM, with title, Investigation of Cytotoxins from Malaysian Cobras, Naja sumatrana and Naja cautia as a potential anti-neoplastic agent. Congratulations, Ms. Ho. Next, Poster 225, Ms. Ka Cheng Wong from UM, with title, Proteomic Analysis Reviews Protective Role of Melatonin in tumor-prone Drosophila melanogaster little giant larvae mutants. Congratulations, Ms. Ka Cheng Wong. Next, poster 223. Ms. Christina Sok Cheng Chong. All right, from UCSI, with title Antibiotic Resistant Patterns of WHO Priority Pathogens Isolated from Acute Pharyngitis Patients in Private Primary Care Clinics. Congratulations, Ms. Christina. All right. Next is the molecular biology category. There's only one winner for category molecular biology undergraduate. The best poster presenter for category molecular biology undergraduate goes to poster 108, Ms. Kachana Apalasamy from AIMS, with title Overexpression and Purification of Actinobacter baumannii HFQ protein in BL21 cells. All right. There are two winners for category molecular biology postgraduate. Okay. Best poster presenter for category molecular biology postgraduate goes to poster 269, Mr. Mingwei Liu from AIMS, with title Transcriptome Analysis of Virus Host Interactions between the lytic bacteriophage DSHS1 and the soft rot macagen Dickeya chrysanthemum. Congratulations, Mr. Ming Wei Liu. Next is poster 206, Mr. Muhammad Ashraf Muhammad Amnan from UM, with title Tandem mass tag-based quantitative proteomic analysis reveals the leaf response to drought in Pandanus amarylifolius. Congratulations, Mr. Muhammad Ashraf. Okay, finally, the last award for category professional. Best poster presenter for category professional goes to poster 325, Dr. Paley Angeli from MPOP, Malaysian Palm Oil Board, with title Oil Palm Tissue Culture System for Artificial Inoculation Study of Ganoderma boninense. Next, poster 331, Assistant Professor Dr. Eugenie Sin Tan from UCSI, with title Pharmaceuticals and Personal Care Products, Residuals in Surface Water Bodies from Selangor, Malaysia, Occurrence and Environmental Risk Assessment. Next, poster 314, Professor Dr. Lau Yiling from UM, with title Real-Time Reverse Transcription Loop-Mediated Isotomal Amplification for Rapid Detection of SARS-CoV-2. Finally, poster 324, Ms. Susanna Mustafa from MPOP, title Preliminary Analysis of QTLs Associated with Yield Components in Oil Palm through Association Mapping. Congratulations to all the winners and to all presenters participated in this 3 minutes poster presentation. All of you did very well. Let's give everyone a big round of applause of virtual claps. Right. All right. Before moving to closing ceremony, we MSMBB would like to extend warmest invitations to all participants to be a part of MSMBB family. You can go to MSMBB website www.msmbb.my or scan the QR code here for more information about MSMBB. Now, coming to the end of this conference, I would like to call upon Professor Dr. Lau Yiling, President of Malaysian Society for Molecular Biology and Biotechnology, to deliver her closing address. Prof. Lau.
Finally, it's the end of our conference. I would like to thank you all for joining the 4th International Conference on Molecular Biology and Biotechnology. Your presence today shows a strong commitment towards promoting scientific advancement, which is the main aim of Malaysian Society for Molecular Biology and Biotechnology. MSMBB is a non-profit organization that was established in 1988 with the objective to promote communication among scientists in the field of molecular biology, cellular biology, and biotechnology in Malaysia and worldwide. We organize yearly conferences, scientific meetings, seminars, lecture series, competitions, and workshops. In the spirit of creating awareness and empowering communities through science, MSMBB has also organized MSMBB TikTok this year, and these are the winners. Another significant accomplishment of MSMBB is the introduction of 2 prestigious awards for scientific achievements. We are so proud to have our 2 awardees for this year. MSMBB also provides EduSTEM grant to support activities which align with UNESCO's Sustainable Development Goals, such as to improve the quality of education, life, health, and well-being. On top of that, we also own our own Scopus Index journal, Asian Pacific Journal of Molecular Biology and Biotechnology. ICMBB is a yearly conference run by the Society. I would like to express my appreciation to the organizing committee of 4th ICMBB for making this event a success. In particular, to Dr. Amira Amil for leading the team, the organizing committee, Dr. Wang, Dr. Tan, Dr. Pui, Dr. Arun, Dr. Kanti, Dr. Adeline, and the rest of us. A job well done! A big thank you for all the keynote, plenary, and invited speakers for sharing their expertise with us. For all the participants, I congratulate you all for your commitment and active participation, and I wish you all the success. Special notes of appreciation to our sponsor for their financial supports: Nanogene Solutions, Research Instruments, Strict Scientifics, Analysa Resources, Impaco, NextGene, XCore, and all of you. Without your financial support for this event, it would not have been viable. I look forward to continuing beyond this event the very stimulating and instructive exchange of knowledge that we had for the past 3 days. I trust that you will take home further the inspiration on how to move molecular biology and biotechnology forward. Thank you, and stay safe.
This is the end of the conference. We'll see you in ICMBB 2022. Thank you, everyone, and stay safe. Bye.
Bye-bye. And I'd like to thanks for our committee as well. Thank you very much. Very nice conference.
Thank you. Thank you.